one in graduate education. So I think that David is a great example of someone whose research spans and blends actually ideas from disparate fields. And I think that his research can be classified in sort of two major research foci. One on the evolution of phenotypic plasticity, and especially the role of competition in Spadefoot toad development, where he's developed an amazing system where different environmental factors can actually lead to the development of omnivore versus carnivore morphs. Hopefully we'll talk a little bit about some of that today. He and his wife Corinne actually had a book on competition and the origins of diversity called Evolution's Wedge that was published in 2012. And his other major research focus is on evolution and development of close-knit mimicry, which of course is a topic near and dear to my heart, where I think his lab has been the biggest and most influential contributor to the experimental evidence for close-knit mimicry over the last 10 years. So without further ado, David.
Thank you, Allison. Is this working? Okay, great. And thank you, Allison and Dan, for inviting me to be here today and for being such terrific hosts. And thank you guys for coming today. It's really been a blast being here. This is, Michigan is one of those like amazing departments for ecology and evolution. Maybe some of you graduate students don't realize this, but throughout my career, this has always been like one of the just sort of key foci for the field. And so it's a real pleasure to be here. So what I want to talk about today is I want to talk about some work that I've been doing on looking at the possible contribution that a phenomenon known as developmental plasticity might play in both evolutionary diversification as well as in innovation. And so let me just begin with just this slide and just note that one of the central challenges that we face in evolutionary biology is explain this. That is to understand why there's so many different kinds of living things, so to explain diversity, but also to understand where the distinctive features that characterize different kinds of living things come from. So where do things like limbs and eyes and flowers and so forth come from? So in other words, where do novel traits or where does innovation come from? Now, traditionally, I think evolutionary biologists have sort of focused on these issues by examining the ecological and the genetic forces that are thought to promote diversification and innovation. And in contrast, I think it's safe to say the role of development has been relatively understudied. And yet it's becoming increasingly clear that a key feature of development, its tendency to be adaptively responsive to changes in individual organisms' environment, might play a critical role in both diversification and in innovation. And it's this topic that I want to focus on in my talk today. So I'll be talking about a phenomenon known as developmental, or what's sometimes referred to as phenotypic plasticity. And I'm going to define it broadly as the ability of an individual organism to alter its phenotype in direct response to changes in its environment. So for example, consider these monkey flower plants. So monkey flower plants in certain populations will produce either this open flower or this closed flower depending upon soil moisture. In the middle are tiger salamander larvae, and tiger salamanders, as with many animals, will produce either a small-headed morph or a large-headed morph depending upon prey size. And then on the far right is my favorite example of developmental plasticity. These are water fleas or Daphne, and these guys are actually clones of each other, so they're genetically identical. And what's happening here is that this individual on the right has been exposed to chemical cues from a predator that the individual on the left was not exposed to. And so you see it develops this distinctive helmeted form that's thought to render it less susceptible to predation. Now you can see from this slide that developmental plasticity is taxonomically widespread. And indeed, I would suggest that it's really a defining feature of living things. But you can also see another really important point from this slide, which is that developmental plasticity potentially provides a mechanism whereby you can get very large-scale phenotypic variation being produced from a single genome. And indeed, this variation is on the order of that which you typically see between different species. And for this reason, plasticity has long fascinated, but at the same time perplexed evolutionary biologists. And so this is the central question that I want to focus on in my talk today, which is what is plasticity's role in evolution? Now, I think it's safe to say that if you were to ask most evolutionary biologists what role plasticity might play in evolution, most would probably say it probably plays little, if any, role. And I think the reason for this can be sort of traced back to ideas such as this. And this is a quote from Dept, who is one of the founders of our sort of modern synthesis of evolutionary biology. And when talking about plasticity, Dept says, consider such an acquired trait as big muscles strengthened by exercise. Its inheritance would require that some products secreted by the muscles change the nucleotide sequence or number in the DNA chains of some genes. Such changes are unknown and seem quite improbable. So in other words, what Dubchansky's saying here is because we cannot envision a way in which acquired characters that are, that bring about plasticity can then be transmitted across to the next generation. Therefore, this phenomenon should play little, if any, role in evolution. Now, at the same time, there have been a number of researchers for well over 100 years that have actually suggested that plasticity might, in fact, play a critical role in evolution. And these ideas, I think, can be best sort of captured by this person. This is Mary Jane West Eberhard, who's a proud alum of U of M. And in her book, Developmental Plasticity and Evolution, she makes this provocative statement where she says most phenotypic evolution begins with environmentally initiated phenotypic change, in other words, plasticity. Gene frequency change follows as a response to the developmental change. And then she concludes by making this even more provocative statement that genes are followers, not necessarily leaders in phenotypic evolution. So she's taking the position, again, that was espoused by a number of researchers back through the decades, that plasticity, rather than playing no role in evolution, was actually playing a leading and often decisive role in adaptive evolution. It's these ideas that I want to examine in my talk today. So what we're going to be talking about today is something that I'm going to just refer to as the plasticity first hypothesis for evolution, of adaptive evolution. Now I want to just say right here, there are many ways in which plasticity might affect evolution, and we're going to touch on a couple of those ways today. But we're mostly going to focus in on this idea that environmentally initiated change might actually play a leading role in both evolutionary diversification as well as in innovation. And I want to illustrate these ideas with a sort of simple set of cartoons. So let's begin with the population, and I'm showing these as tadpoles because that's what I study, tadpoles. And let's imagine that it's a genetically diverse population. So we're indicating here as the different colors are meant to signify different genotypes. Now let's imagine our population of tadpoles here experiences some novel environment. I'm indicating the exposure to novel environment by the gray background here. Now exposure to a novel environment is something that's going to happen to all lineages at some point in their evolutionary history. And it's also important to point out that exposure to a novel environment is typically very stressful for organisms, right? So one way in which, probably the main way that organisms respond to the stress that's associated with novel environments is to use their inherent developmental plasticity. So think about in mammals. When we get cold, we shiver. When we sweat, or when we get hot, we sweat. So those are both examples of adaptive plasticity. So what we see here then is that novel traits begin to be induced by this change in the environment. And that's indicated here by the little dashed lines. But here's the key point. The key point is that typically different genotypes are going to respond differently to the same change in the environment. So for instance, in our hypothetical example here, the blue genotype changes by producing this sort of bulgy body form, let's say. The brown one goes the opposite direction. It becomes skinnier. The green one is sort of in between, and then the yellow one doesn't change at all. And in fact, in every natural population that I'm aware of where this has been studied, researchers have found this sort of underlying genetic variation in the degree to which individuals respond to some environmental cue or whether they even respond to that cue in the first place. So once that variation, which was formerly cryptic, once that cryptic genetic variation then gets revealed, then selection can begin to act on it, and it can do so by disfavoring the maladaptive traits, genotypes, for that new environmental circumstance. So let's imagine that this bulgy body form now is the adaptive form for this new environment. So selection could favor it, but it could also start to refine that trait. So it could start to produce a better and better version of that trait, and that's indicated here by just the larger body size. Now, if you have a situation like that where selection starts to refine this trait, where everybody begins to produce that new form, that bulgy body form in this case, in some new environment, and then you've got the ancestral phenotype back in the ancestral environment, this is what's referred to as a polyphenism, where you get environmentally initiated alternative phenotypes. But it's also possible that over time, selection might actually favor the fixation of that one phenotype so that they lose the ability to be responsive to the environment. They become fixed in that population, even if the population were to experience the ancestral environment. And this is a phenomenon that has been dubbed genetic assimilation, where you go from what was formerly an induced trait to one that loses that induced ability and becomes expressed constitutively. But the point I want to make here is that whether you have a polyphenism or genetic assimilation, the result of this process is that you may end up here with a novel trait. So a new trait that is produced that was initially expressed only in occasional response to some environment, it's become refined over time. But you can also see that if you have within two populations where one selection is favoring one phenotype over here and a different phenotype over there, you might also expect to get the buildup between these populations of reproductive isolation. And of course, that's a potential critical stage in the process of speciation. So not only might you get a novel trait, but you might end up with a new species through this process. All right, so the point here is that through this sort of process that I've laid out here, then plasticity might play a critical role in two major events in macroevolution, the origins of novel traits and even potentially the origin of new species. So the question is, what are the reactions that people have had to these ideas? And as I said, some of these ideas have been around for a long time. And you know, these are just two quotes that I pulled out recently. So here's a chapter by Doug Fatuma, who's of course one of our leading evolutionary thinkers, and he says considerably more research on a range of taxa and characters will be needed before we can judge whether or not plasticity often leads adaptation. And then here's another quote from a counterpoint that appeared in Nature a couple of years ago, again by some of the leading thinkers here. And the point they're making is the best way to elevate the prominence of genuinely interesting phenomena such as developmental plasticity is to strengthen the evidence for their importance. So the point here is that we've had this theory for a long time. The question is, how do we go about evaluating this theory? And that's what we've been trying to do in my lab. And so with one of my current graduate students, we have a paper that just appeared this week online. It's going to come out this summer in Tree. And the goal that we're trying to lay out here is to come up with a scheme whereby we can begin to evaluate these ideas in natural settings and natural systems. And so again, our point here is that we're not advocating one position or the other. Our goal is to provide a means by which plasticity and adaptive evolution can be evaluated or assessed. And so one of the things we try to do here is we try to say, okay, like any evolutionary hypothesis, we want to develop some critical predictions and ask, well, if this idea is right, what kinds of data would we expect to find that are that allow us to either support or reject this hypothesis? And so there are three predictions that I'm going to talk about today. And in particular, if we are hypothesizing, if we're looking at some trait in some group of organisms, and we're wondering, did plasticity precede and even facilitate the evolution of that novel trait, then we should find that three conditions are met. First of all, of course, most fundamentally, we should find evidence that that trait is produced at least under certain environmental circumstances in some ancestral lineage, right? So there should be ancestral plasticity. So ancestral species should express the trait through plasticity. And you'll see why I put the quotes around that in just a moment. Secondly, we should find some evidence that novel environments in that ancestral lineage uncover cryptic genetic variation. Remember, it's the uncovering of that cryptic genetic variation that really provides the fuel for this process to unfold. And then thirdly, we should find some evidence that trait expression has undergone adaptive refinement. So obviously we expect to see some evolution of both the sensitivity of that trait to environmental circumstances, whether it becomes a fully developed polyphenism or genetic assimilation where it becomes more canalized. But we should also find that it's producing a better and better version of that trait as it's exposed to selection more and more frequently. And so what we do is we lay out a scheme in our paper whereby in a fairly simple way you can actually evaluate all these predictions pretty much simultaneously. And so the first step is to basically identify your players, right? And so again, I've shown these as little tadpoles here, but they could be anything. And let's imagine that we're focusing in on this lineage out here. And this is the lineage that produces our novel phenotype that we are interested in asking, did that novel phenotype start out as a conditionally expressed trait in some ancestral lineage? So in this lineage, lineage 3, again, we got that bulgy body form, but let's imagine this is now fixed, it's canalized. So we might expect in this sort of scenario like what I outlined a moment ago, you might expect to find some ancestral lineage where that phenotype now is produced, but it's produced only under certain environmental circumstances. So you see that phenotype being triggered in response to some change in the environment. And eventually you want to go back here to some lineage, let's say this lineage 1, where you have a phenotype that's quite different from your derived phenotype, your bulgy body novel phenotype over there on the far right. But this is a lineage that normally will not express that new phenotype under the circumstances, the environmental circumstances that that lineage normally experiences, all right? And so once you identify your players, you can do the following kind of design. This is basically just a split, common garden design. So you can take your three lineages and you want to rear them in the ancestral environment. But you also want to rear them in the novel or the derived environment. So this is the environment that that novel phenotype is normally associated with. And so what you're asking here is you want to find out, is there any evidence of pre-existing plasticity in that ancestral lineage over here? And so do you start to see some rudimentary version of your novel trait being produced in response to exposure to the novel environment? But you also want to look at the reaction norms. So basically you're taking different genotypes, different SIB-ships, and you're rearing them both in the ancestral environment and in the novel or the derived environment. And you want to look for things like this. So for instance, if you were to find this pattern where more variation is revealed in the novel environment, that would be suggestive of cryptic genetic variation being uncovered in that lineage. You might also want to look for evidence that a polyphenism has evolved. So if you find that these reaction norms, which is what these curves are called, if they all sort of collinear, but they're at an angle like that, then that would be suggestive of a polyphenism, a novel polyphenism. And then lastly, you might want to look for evidence of genetic assimilation. So if these lines are all flat like that, meaning they're unresponsive to changes in the environment, then that would be suggestive of a genetic assimilation. And then the last thing you would want to look at, ideally, is you'd also want to ask, is there evidence that this evolution, the change in responsiveness to the environmental cue, is it producing a more adaptive version of that trait? And so for instance, one way to look at this is to then take the canalized phenotypes and basically put them in direct competition or put them in the same common environment, the derived environment, with the phenotypes or with the lineage where they're only producing that phenotype under certain circumstances. And so you would predict here that the canalized phenotype should basically have higher fitness in this novel environment than the induced form over here, because this form is exposed to selection more frequently. Right? So a number of researchers have begun to do this sort of thing, and I realize you're not going to be able to read this table. I just put it up there to emphasize the point that there's evidence now supporting various predictions of this plasticity first hypothesis, ranging from plants to various invertebrates to fish and amphibians and snakes and even some birds. So the evidence is starting to come in, basically, and it's starting to support at least aspects of this hypothesis. So what I want to talk about today, though, is I want to talk about some work that we've been doing in testing this plasticity first hypothesis in a natural population and an organism in a natural environment. So what we do to do this work is we head out to the southwest each summer. So these are the Chiricahua Mountains in southeastern Arizona. And we wait for this to happen. Some years this doesn't happen at all, but hopefully if we're lucky, you see the summer rains come. And we wait for this to happen. We wait for the rains to form water in low-lying areas like these temporary pools. And then we wait around for nighttime and we wait for this to happen. So these are spadefoot toads. So the desert basically comes alive with these toads. So, oops, I went forward one. I didn't mean to do that. So this guy right here that's making that quacking sound, that cute guy, that's the plain spadefoot, Spiabomifrons. You hear two other calls in the background in that same pond. So the call that sounds like running your finger down a comb is the Mexican spadefoot, Spia multiplicata. And then you hear the sort of bleating of a lamb in the background, that sort of wah, wah. That's the Couch's spadefoot, Scaphiopus couchi. So you can get all three of these species potentially breeding in the same ponds. And so the reason we're interested in studying these guys is that they display a spectacular form of developmental plasticity in their tadpoles. And so if you go into these ponds and sample the tadpoles, you find some tadpoles that look like this guy up here. This is like a sort of standard tadpole that you'd see anywhere in the world, including here around Ann Arbor. And so these guys have sort of a football-shaped body, kind of a narrow head, and nothing too exciting about that. And so I'm going to call this the omnivore morph. And then you'll find other tadpoles in the same pond that have a very different morphology. And they've got this big flat head. And if you look at them in more detail, they've got greatly enlarged jaw muscles. They've got a greatly enlarged and serrated keratinized mouth parts here on the front end. And you'll see a picture of that in just a moment. And then if you look at their gut, they've got a really short gut. So their gut is very, very much shorter than these guys. And I'm going to refer to these guys as the carnivore morph. Now we're dealing here with an example of developmental plasticity. Specifically, we're dealing here with an example of diet-induced plasticity. And you can demonstrate this by taking a young tadpole and asking, did that focal tadpole ingest large animal prey early in its life? If the answer to that question is no, then it invariably develops into this omnivore morph. If the answer to that question is yes, and the large animal prey we're talking about here, I should have mentioned, by the way, like these anastrachan fairy shrimp, or even other tadpoles, they're really cannibalistic. If they do eat those things, then there's the potential that they might go on to produce that carnivore morph. And I want to stress that the potential they might produce that carnivore morph, because in every population we've sampled, as I indicated earlier, as other people find in all kinds of other animals and plants, we find there's genetic variation in ability to respond to that cue. So some genotypes never produce carnivores, some produce lots of carnivores, right? So as you might imagine from their names, these guys occupy different ecological niches. The omnivore morph is a dietary generalist. They eat on plants, detritus, and small crustaceans. The carnivore morph is a dietary specialist. So they specialize on these large anastrachan fairy shrimp. And they said they will occasionally also go after and eat another tadpole, but that's less common than just eating these shrimp. Now I want to just say a few words before I dive into the plasticity part, just talk about the evolution and the development of these kinds of alternative morphs, because there's actually some interesting points to be made here. One point I want to make is that, when you find 2 or more morphs in a system like this, you think there's something kind of interesting going on, because our caricature of how selection should work in a population is that it should tend to favor some optimal phenotype in that population and eradicate all other phenotypes in that population. So when you see 2 or more morphs in a population, you begin to think something unusual is going on. And of course, one possibility is that selection doesn't always have to act that sort of stabilizing way that I just showed, but it could also act in a disruptive fashion, where you have selection favoring extreme morphs and disfavoring intervening intermediates between those 2 extremes. So we were interested in asking, is there evidence for disruptive selection in our system? So we went into these ponds, and we went into dozens of ponds now, and we sampled the tadpoles. Here you can see a carnivore, and these other dark guys there in the background are omnivores, and there's one of the fairy shrimp you can see up there. And so we characterized the overall shape variation in this system by developing what's called a morphological index. It's just a composite shape variable. And then we needed a proxy for fitness. And in this case, our proxy for fitness is body size. And that's a good proxy for fitness in this system, because in this system, as in many others, bigger is fitter. So larger tadpoles have higher survival to metamorphosis. Larger individuals at metamorphosis are also larger at maturity. Larger males have higher mating success. Larger females, higher fecundity. And so we're plotting fitness here against this morphological index. More carnivore-like is that way, more omnivore-like is this way. And these are just the data for 6 different ponds. Again, we've done this in about 40 ponds now. And then the little dots there are each individual tadpoles. And what you can see here is that in all 6 of these populations, there is indeed evidence of disruptive selection. There's an intermediate fitness minimum. And in fact, in 5 of these 6, there's significant disruptive selection. This is the exception there, and it's probably a power issue. So what we find here is intermediates are indeed disfavored in this system. So the question now is, what is it about intermediates that renders them having lower fitness? And the answer seems to be the following. So what we find is that omnivores have this really long gut. So here's, you can see this gut, and a little tiny tadpole can have a gut that maybe is a meter long in total length. And so what that means is that they're really good at assimilating the low nutritive quality diet that the omnivore feeds on. So the detritus, which is junk on the bottom of a pond, and the plant material. The carnivore, by contrast, has this really short gut. So they're not at all good at utilizing detritus. In fact, they starve to death. But they have these keratinized mouth parts. They have these large jaw muscles. So that enables them to be really good at capturing and consuming the large animal prey, like the large fairy shrimp and the occasional tadpole. So if you're in between these 2 extremes, then you kind of aren't very good at using either of those 2 resources. And so we think that's why then the intermediates are not favored in this system and why the extremes are favored. The main point I want to make from this slide, though, is that neither morph is really intrinsically better than the other. I mean, they're basically adapted for different kinds of resources. But the other thing I want to just talk about very, very briefly is the development of alternative phenotypes like this. And traditionally this has been sort of a black box. How do you get a single genome producing 2 such dramatically different phenotypes as we see here? And as people began to think about this in other systems, they realized what must be going on here is you must have different genes being expressed in response to different environmental cues. So one set of genes is being expressed in response to some environmental cue, and some other set of genes is being expressed in response to some other environmental cue or the absence of that first cue. So we look for evidence of differential gene expression in our system, and we've thus far found several dozen genes that are indeed differentially expressed between these 2 morphs. And I just want to summarize the data here because it's kind of interesting. So the genes that we find thus far that are expressed more highly in carnivores than in omnivores have been implicated in functions such as muscle development, keratin production, and digestion of proteins. And that all makes sense to me because remember, these guys have these really large jaw muscles. They've got these large keratinized mouth parts. But they're also eating a diet like these fairy shrimp that are really, really rich in protein. The genes that we find thus far that are expressed more highly in omnivores than in carnivores have been implicated in immune function, digestion of fats, and digestion of starches. And again, that makes sense because the omnivore is feeding on detritus. They're feeding on, again, just junk on the bottom of the pond. And a lot of that junk is just tadpole poop. So presumably their immune system is really being challenged. But of course, their diet is really high in fats and starches. So these genes are what we hypothesize could be under the targets of selection that we're going to be talking about today. So with that background in mind, the background to the general questions, the background to the study system, I want to turn back to this question of, well, what is plasticity's role in evolution? And for the remainder of the talk, I'm going to break this down into two specific questions. First of all, we're going to ask, what role does developmental plasticity play in innovation? So specifically, does developmental plasticity precede and facilitate the origin of novel complex traits? And then we want to look at its role in diversification. Specifically, does plasticity precede and facilitate the origin of new species and trait differences between them? And so we're just going to take these two questions in turn. So let's begin with this idea that the origins of novelty, the origins of novel traits might often begin through this sort of plasticity first scenario that I outlined earlier. So how do we go about testing those ideas? So we're going to specifically zero in on this carnivore. And again, you can see the guts here. This is it's really the guts that we're going to look at. So here's the omnivore with its really long gut. Here's the carnivore with its short gut, short-coiled gut. This is a very unusual feature of a tadpole. Most tadpoles do not have this sort of really short gut. This is very adult-like. Adults are carnivorous and they have short guts, but tadpoles tend to not have that. And so what we want to know is, did this novel morph, specifically this short-gutted form, did it arise through a sort of plasticity first scenario, as we saw earlier? So as I indicated earlier, in order to test that idea, we have these three predictions that we want to basically evaluate in our system, beginning with this idea of whether the ancestral species express the trait through plasticity, whether there's cryptic genetic variation being revealed, and whether there's evidence that the trait has undergone adaptive refinement. So the first thing we do, is, as I indicated earlier, is we want to identify our players in our system. And so we start out with what's called an ancestral character state reconstruction, where we're basically mapping on a phylogeny the distribution of taxa that have both those morphs present, so both the omnivore morph and the short-gutted carnivore morph, and those are shown here in the black circles, and then those taxa that produce only the omnivores. And so here's the modern genus Spia, this is the modern genus Scaphiopus, and then I just say over here other frogs. These are clearly not all frogs, but like these are Xenopus over there and so forth, right? And so what you can see, first of all, from this character state reconstruction is that, you know, this is a novel trait, right? So it's restricted only to this one clade up in the upper left, that short-gutted carnivore morph. It's only being produced in this modern genus Spia. So what we want to do now is we want to take this ancestral form right there and use it to test, to start to evaluate our first two predictions. But of course there's a problem, and that is that ancestor is not with us anymore, right? It's extinct. So we have to use a proxy for that ancestor form, and that's why I put the ancestor in quotes earlier. So in our case, what we choose for a proxy is an extant species that's in good position phylogenetically, and that's the Couch's spadefoot. Now, Couch's spadefoot is a good proxy for this ancestral Spia down here that we suspect only produced omnivores, because the Couch's spadefoots only produce omnivores. This is one of its tadpoles. They don't produce the carnivores at all. But they're also in a good position because, as we heard earlier, they're found in a lot of the same populations with some members of this genus Spia. So remember, we heard the Couch's spadefoot calling in the same pond with the Spia bomifrons and the Spia multiplicata. So we take our Couch's spadefoots into the lab and we begin to go through our three predictions. So we start with answering this question: does the ancestor proxy species, Couch's spadefoot, express the trait, the short gut like you see on a carnivore, via plasticity? That is, under only certain environmental circumstances. So we take Couch's spadefoots and we create different SIB-ships in the lab, and then we split these SIB-ships up into two different groups. So half of each SIB-ship is reared on detritus, and that's their standard diet. That's what they feed on in the wild. The other half of each SIB-ship we expose to this novel derived diet, right? So the shrimp. And this is not a diet that they would normally feed on in the wild. Now, I want to be clear here. Of course, they're encountering shrimp. They're just not eating them. But we're forcing them to eat on those shrimp, or they starve. I mean, they don't get anything else other than those shrimp. And then what we do is, as our response measure here, is we use gut length as our proxy. So what we're looking at here is gut length of each SIB on detritus and on shrimp. So these lines are connecting different SIB ships, and then different lines are connecting siblings, and then the different lines are different SIB ships. And what you can see is that when you look at what's happening on the shrimp diet relative to the detritus diet, they are indeed showing plasticity. They're producing overall slightly shorter guts when they eat the shrimp than when they eat the detritus. Now I want to be clear here, this is nowhere near the magnitude of the differences in gut length that you would see in SPEA, right? So SPEA would be down here in the basement or something, right? They produce really short guts. But the point is, they're producing a novel trait here only through exposure to this novel environment or this derived environment, and it's in the direction that we see the canalized trait. So we provisionally accept our first prediction as being satisfied. Now the second prediction was to answer this question, whether we see novel environments uncovering cryptogenic variation, and you can kind of see evidence for that just graphically over here. You see a lot more variation revealed in the shrimp diet than on the detritus diet, but we can quantify that variation through a measure called heritability. So this is just basically a proxy for the overall phenotypic variation that can be explained by genetic variation. And for those of you who know, this is actually, we're using broad sense heritability here, so there's some limitations with that. So this is a value that ranges from 0 to 1, 0 meaning no heritability. So we look on detritus, there's no evidence of genetic variation, cryptic variation being revealed. But if we look at what's happening on the shrimp, there is in fact significant heritability, suggesting that there's significant genetic variation being revealed on that diet. So we conclude that our second prediction is satisfied. So our third prediction involved answering this question, which was to ask, has trait expression undergone adaptive refinement in more derived species? Now this is a little bit trickier question to answer. So what we chose as our proxy here was to look at a measure called gut cell proliferation. So what you're looking at here are two different tadpole guts that are stained for cells undergoing mitosis. And you see there's many more mitotic divisions up here than down here. So what we find is that if you feed a tadpole a diet that it survives and grows well on, they tend to show much higher levels of cell proliferation than if you were to feed a tadpole a diet that doesn't survive and grow well on. So this is basically a proxy for how well adapted that tadpole gut is for a specific kind of food resource. So we look at gut cell proliferation in response to different diets in our ancestor proxy species, Couch's spadefoot. But remember, we're looking for evidence of adaptive refinement, so we have to also pull in two of our derived species, and these are two members of the genus Spea, the Mexican spadefoot and the plain spadefoot. And for reasons you'll see in just a moment, the plain spadefoot is the one that produces much higher proportions of carnivores in natural populations, all right? And so what we're doing is we're looking at gut cell proliferation in response to eating on shrimp, and that's shown in the maroon bars here, in response to eating on detritus in blue, and then we have an unfed control group over here in gray. And I want to draw your attention down here first to our ancestor proxy species. So if you look at what's going on here, they show no more cell proliferation when they're fed shrimp than in the unfed control group. So at least from the point of view of their guts, what their guts are telling us is it's like they're not even recognizing this as a food resource. And in fact, some of these SIB ships actually starved to death before, I mean, they weren't even eating these shrimp at all, they just starved, right? So they're not utilizing this shrimp, and their guts are not presumably adapted to utilize the shrimp. And if you contrast that to the situation up here to the plain spadefoots, they show significantly higher proliferation on shrimp than on detritus than an unfed control group. So at least these data are consistent with the notion that trait expression has undergone adaptive refinement towards the evolution of this trait. So what we hypothesize went on here in this system is that we started out ancestrally with a population that was like modern day Couch's spadefoots. They produced only these omnivores. Now these guys were faced with a problem though, and that is that for the last 10,000 years or so, we know the Southwest, where these guys live, has been drying out. And so the problem was, how do you get out of these ponds before they dry up? Now, one way for a tadpole to speed up its development is to get a more nutritious food resource, right? And so sitting out there was this abundant ecological opportunity in the form of these anastracan furry shrimp. Nothing else is utilizing these shrimp in these ponds, right? And so there's a really high nutritious food source. So presumably a few individuals had a tendency to start eating those shrimp. You know, that gave them a survival advantage. They were more likely to get out of these ponds before they dried up, and they took with them those tendencies to go after those shrimp. But then over time, you also started to see the revealing of these other kinds of traits, and selection started to refine them. So the large jaw muscles, the keratinized beak, and of course the shorter gut. So it might have started out as just slightly shorter, got shorter and shorter as selection started to refine that trait. Now eventually we did not see the fixation of a carnivore morph in this system. In other words, we didn't go towards genetic assimilation of the carnivore morph. What we see in this system instead is the evolution of a polyphenism, where we get these two morphs being maintained as sort of a developmental switch, right? And we think the reason for that, as we saw earlier, is neither morph is intrinsically better than the other, right? Now, we're going to see in the next section there are, in fact, populations where we see fixation, near fixation of this morph, and there are populations where we see near fixation of that morph, right? And so we're going to see situations where that does occur, but in most populations you get this sort of developmental switch. So the bottom line here is that we think we have pretty good evidence in this system for this sort of plasticity first scenario maybe playing a role in the origins of this novel carnivore morph. And we think this kind of scenario could have unfolded in other systems as well. So what I want to turn to now for the remainder of the talk is to look at the other question that I posed, which is what role does plasticity play in the origins of diversity? And again, we want to ask, is it possible that new species even and trait difference between those species could have started out as environmentally initiated change? So to introduce this part of the section, though, I want to just begin by asking, well, what do we know about the origin of species and species differences? And of course, I'm going to overly simplify things here, but I want to turn back to Charles Darwin. And you know, Darwin obviously thought about this question, and for Darwin, the critical feature was an ecological interaction. And you know, Mark Ridley's pointed out if Darwin hadn't discovered natural selection, he'd be famous as the father of ecology, because he really got us thinking about how ecological interactions can play an important role in natural communities. And that specific ecological interaction that Darwin keyed in on was competition. And he said, look, all organisms face recurrent competition for scarce resources. And that competition is going to favor individuals that somehow differ from their competitors. And then he said over time, interacting populations should diverge from each other. He called these varieties and so forth, until eventually they become separate species. And so this was his sort of scheme of how this would unfold, was this figure that he presented in the Origin of Species. He referred to this process as divergence of character. And so what he's talking about here is how selection to minimize competition can push things apart in morpho and ecological space. And again, we can sort of cartoonize this idea. And let's imagine that we've got two populations of some hypothetical bird species or seed-eating birds. And these are the different seed sizes they feed on, and this the beak sizes. Now, if they were to overlap with each other in resource use, of course, we would expect individuals in the extremes of these distributions to be favored, all else being equal, because they would have fewer competitors with which to contend for those resources. And so over time, that should cause these populations to diverge from each other. And eventually, you might expect to get reproductive isolation building, and they might become separate species. So this process, whereby you get trait evolution arising as an adaptive response to minimize resource competition, is something that we now know as character displacement. And so the question is, what role does plasticity play in character displacement? Well, we have a really good opportunity to study character displacement in the Spadefoot system, because we could take advantage of a signature that has long been hypothesized to, that you could go out and look for character displacement with. And that's the idea that if two species have in fact been undergoing character displacement each other, you should find a distinctive pattern where they show exaggerated divergence in traits associated with resource use, where they co-occur, but they could be virtually identical to each other in that same trait, where they occur in the absence of each other. So in allopatry versus in sympatry. So we have a really good opportunity to look at this, to look for this signature of character displacement in our system, because in a fairly small geographical area in southeastern Arizona and southwestern New Mexico, we get all three kinds of populations occurring nearby each other. So let me explain what I mean by this. So these yellow dots are locations of ponds, breeding ponds, where we consistently find only Mexican spadefoots breeding. So these are allopatry for the Mexican spadefoot. These little dots in blue here are ponds where we consistently find another species breeding by itself, and that's the plain spadefoot. So that's allopatry for the plain spadefoot. And then lastly, here in the green stars, and up here also, these are ponds where we consistently find both species breeding together, right? And so these are sympatry. So we can go into these different ponds and we can sample the tadpoles and look at their resource use. And the way we look at resource use here is to look at the proportion of carnivores produced, because remember, that's a resource use trait, basically. And so here are the data for natural ponds. So this is proportion of carnivores. This is for the Mexican spadefoot, the plain spadefoot. And in each case, allopatry is on the outside, and then the green bars in the middle are sympatry for the two species. And you can see that when they are found in the absence of each other in allopatry, both species are producing about equal intermediate frequencies of both morphs. So overall, there's no significant differences here between the two morphs. They're producing both carnivores and omnivores in allopatry. But if you look at sympatry, you find that the one species, the Mexican spadefoot, is producing almost all omnivores, almost no carnivores at all. Whereas the other species is doing the opposite. It's producing almost all carnivores, very few omnivores, right? So those are the data from natural ponds. The question is, what happens if we bring these adults in the lab and we rear them up? We find the exaggerated divergence. So we rear them up in a common environment. We find those differences hold up, right? And so what I'm not going to talk about today is our evidence that this divergence does indeed reflect character displacement, because that's a whole story in itself. Instead, what I want to turn to is what role might pre-existing plasticity have played in facilitating this kind of divergence between populations and between species. And so the question that we're going to look at here is, does plasticity facilitate character displacement in particular, but more generally, does character displacement facilitate divergent trait evolution? Now, the scenario that we've outlined here with the plasticity first scenario was actually sort of anticipated by E.O. Wilson. And E.O. Wilson is relevant here because Wilson was one of the two scientists who coined what I think is a horrible term, character displacement, but he's the one who coined this term, character displacement, in the first place. And in his book, The Diversity of Life, he lays out this short little quote here, this little part here, where he's talking about how he thought character displacement would unfold in natural populations. And listen to what he says. He says, imagine a case in which two species have been squeezed together in the same communities long enough for evolution to occur. When they first came into contact, they were elastic, meaning they were plastic and could diverge in their habits enough to lessen competition. As the generations passed, genetic differences arose and hardened the distinction between the two species. So the point here, and not to argue by authority, but the point here is that he's basically laying out a plasticity first scenario, as we sort of saw it earlier, as his explanation for how character displacement unfolded. Now, we actually have a good opportunity to look at how character displacement might have unfolded in this system, because we essentially have a time machine for studying character displacement. And let me explain what I mean by that. So here are the overall geographical distributions of the Mexican spadefoot in yellow here, and the plain spadefoot in blue here. And then this area of green is where those two species potentially co-occur in New Mexico and surrounding states. Now this down here is the location of the studied populations that we just saw a moment ago, where we've documented character displacement between these two species. Now, what we've discovered is that we've got a number of lines of evidence, a lot of genetic data, but even, believe it or not, museum specimens, to suggest that for the last several thousand years, the one species, the plain spadefoot, has been expanding its range southward and westward into this new ecotone over here, basically into the deserts, right? So it's been coming out of the plains and it's been expanding rapidly southward and westward. As far as we can tell, the other species, the Mexican spadefoot, has been stable more or less during this time. I say it's amazing. We actually have museum specimens from like the 1870s down here, and Geronimo wasn't captured until 1876. So I can imagine some herpetologists out there, dodging arrows from Geronimo and his guys, and the Apaches, collecting the spadefoots. But anyway, we have evidence that, as I said, this one species has been moving southward and westward. So what that means for our purposes today is that the allopatric populations here represent the ancestral state. We've got secondary contact here. And of course, that means then the sympatric populations are derived. But here's the actually interesting twist. In southeastern Arizona, the sympatric populations of the Mexican spadefoot can be thought of as representing recent sympatry, because they've only recently encountered this competitor coming in from the northeast. The other species, the plain spadefoot, can be thought of as being an ancient sympatry, because it's long been overrunning the habitat of the other species, the Mexican spadefoot. So we basically can look at character displacement at these 3 different points in time, or in these 3 different types of populations, and infer how it might have unfolded over evolutionary time. So we're going to look at the evolutionary character displacement beginning with first contact. And so what we do to simulate first contact is we take animals from allopatry, and we take individuals of each species and rear them either with a single conspecific or rear them with a single heterospecific, and then give them limited amounts of detritus and shrimp, and then again we measure the proportion of carnivores being produced. And what you can see here is, once again, they're producing about equal frequencies of carnivores when they're reared with the conspecific, but when they're reared with a heterospecific, these guys from allopatry already had pre-existing plasticity that basically allowed them to sort out these 2 different ecological niches. So we see that plasticity mediates the divergence here on first contact. Now I want to just step back for a moment, because I think this is actually a really important finding that people who study plasticity and its role in evolution maybe ignore a little bit, which is that, think about what we have here. We have these 2 species that normally in allopatry are producing these 2 different ecomorphs, right, that are representing these 2 different ecological niches. When they come together, each of these species is giving up one of those morphs, and that presumably allowed them to stably coexist. This may be the most important influence that plasticity can have on evolution, which is this sort of indirect effect, which allows populations to just basically deal with rapidly changing environments and just stay in the game long enough to, for other things to happen later on, right, to diversify and so forth. But what we want to do is we want to move forward in time and move into recent sympatry, and this is where I think things get really interesting. So what we're going to look at here is the mechanism of character displacement occurring between the Mexican spadefoots up here in yellow versus down here in green. So these are neighboring populations. And so what we can do is we can cross females and males across these different populations. And then what we're looking at here is a behavioral measure of how carnivore-like their tadpoles are. Basically, lower values means you're more carnivore-like in this case, right? So it correlates with how likely you are to produce that carnivore. But the point I'm making, this is the parent source populations, allopatric female with allopatric male, allopatric with sympatric male, and so forth. The point I'm making here, though, is that the offspring always look like the mom's population. It doesn't matter where dad came from. So this got us thinking maybe there's a maternal effect here. So we began to look at this in more detail, and we find there is indeed a maternal effect. If you look at how carnivore-like the tadpoles are, and again using this behavioral measure, and plotting that against maternal conditions, so this is basically a measure of how fat a female is for a given body length. What we find is that females that are in better condition are producing more and more carnivore-like tadpoles. And we think the reason for this is that these females that are in better condition are producing larger eggs that are hatching out as larger tadpoles, more likely to get that shrimp Q that produces the carnivore morph. Now, how would this be manifest in the wild? Well, if you look at a mean condition of females in a population, each of these dots is the population value for all the females in that population. We find that females in sympatry are actually in worse condition than females in allopatry. And it's sort of a graded response. As they encounter more and more of the other species, the plain spadefoots in their population, they're in worse and worse condition. And we think the reason for that is that as you get deeper and deeper into sympatry, the Mexican spadefoot is producing fewer and fewer of these big carnivores. And so we think that's why they're coming out as smaller adults, because they metamorphose at smaller body size. Now we can pull this all together and do the following experiment. So this is where we're crossing females just with males from their own population, but this is when the females are fresh caught. And again, this is more carnivore-like or lower values here. And this is where parents are from allopatry versus sympatry. And in this case, allopatric females are in better condition because they're fresh caught, and they're producing more carnivore-like tadpoles. But now we take those same females from allopatry and sympatry, we keep them in the lab for a while, for about 6 months, we equilibrate them body condition by adjusting their feeding, and then we test them again, and you can see that the differences go completely away. So what this is telling us then is it looks like the character displacement here is just reflecting a maternal effect. So it looks like this maternal effect starts to take over in recent sympatry. Now when you go into ancient sympatry and look at the mechanism in the other species, the plain spadefoot, we can create crosses between the 2 species. There's no evidence of any sort of maternal effect here. They always look like the plain spadefoot. But also what's kind of cool is that if you look at some of these populations of the plain spadefoots in sympatry, they're just coming out as little carnivores. So this is pre-feeding. So they didn't even get exposed to the shrimp Q. So it looks as though they've already, they're already gone towards carnivores. They're just hatching out as little baby carnivores. So it's at least consistent with the idea that differences here are becoming genetically fixed. So what we think has gone on here is you had these two species. When they first came together in initial contact, plasticity mediated the divergence, but that plasticity had consequences for the very next generation. So you had these guys producing omnivores in the Mexican spadefoot, mature as smaller females, produce smaller eggs, that then go on to produce mostly omnivores, and then just sort of perpetuates that trend. And so you get in that recent sympatry, you get this maternal effect that starts to stabilize those differences. So this arose in one generation. This can potentially take over in generation 2. So it starts to harden those differences. And then eventually, we think that these differences become genetically fixed. So it looks like it's consistent anyway with the idea that genetic assimilation has led to the evolution of the character displacement in this system. But to me, it's got this interesting twist, which is it's got this sort of epigenetic assimilation in the middle that's being mediated by this maternal effect. Now, I haven't said anything about speciation. I just want to say a few quick words. So we have in this Mexican spadefoot, these populations that are right next to each other diverge in this ecologically relevant trait in terms of the number of carnivores they're producing, the proportion of carnivores. So we might expect to see the buildup of reproductive isolation between these populations. And in fact, that seems to be what's going on here, because you can cross individuals within a competitive environment and between these competitive environments. And when we do that, we find that between competitive environment tadpoles are inferior competitors to the within competitive environment offspring. So whether you cross sympatry to sympatry, allopatry to allopatry. So that's at least consistent with the notion that you're already starting to get what's called ecologically dependent post-zygotic isolation. But we also find, using indirect DNA-based methods, that gene flow appears to be slightly reduced between these competitive environments relative to that within each of these two competitive environments. And then lastly, we're finding that sympatric and allopatric populations have also diverged in female male preferences. So we're not saying we're seeing speciation, of course, but we're looking at maybe the early stages of reproductive isolation building. So the point here is that developmental plasticity we've seen can facilitate character displacement. Character displacement has long been thought to play an important role in the evolution of reproductive isolation. So by inference, plasticity maybe promotes reproductive isolation. So I just want to conclude with a couple of points. First of all, we've seen that plasticity potentially adds a new dimension to our view of evolution, but also, of course, ecology as well, in terms of allowing populations to stably coexist. And we've seen that plasticity provides a mechanism whereby populations can undergo rapid and adaptive change in direct response to changes in the environment. So potentially it's affecting many individuals simultaneously because they're all being exposed to that change in the environment. And it's possible that plasticity might have these indirect effects, but it might have these more direct effects too, where it might jumpstart phenotypic change in an adaptive direction. And genetic differences, at least in some cases, might actually evolve later, the differences that harden those initial differences. And I just want to end by noting that, of course, the plasticity first hypothesis, as we've seen, it does have some support, but really I think where we're at right now is we just need more tests, more different systems, more kinds of traits, and more different kinds of ecological settings and so forth. So with that, I'll just close with my acknowledgement slide, but also again close by thanking you guys for inviting me in for coming to the talk today. Yeah, Bob. Yeah, it's a great question. I forgot to mention that. So it looks like so we have looked at this now in multiple what we think are evolutionarily independent situations, right? So like in Oklahoma, southeastern Arizona, Texas Panel, it's all going in the same direction. Where Bomafrons is more the plain spadefoot is more carnivore-like, and we think the reason for that is that if you look at plain spadefoots in allopatry and compare them to Mexican spadefoots from allopatry, and of course it's a little bit apples and oranges here, but the plain spadefoots are coming out larger. They also, there's this term personality these days. They have different personalities, right? So behaviorally they're very different. So the plain spadefoots at hatching in these allopatric populations, they're a lot more active, and so I'm thinking they're just, they're better at getting that cue. And so when you put this larger, more active tadpole in with a smaller, less active tadpole, they're getting more of the cue. Now the Mexican spadefoot, you may know this, they actually develop a little bit faster, so they're actually hatching out slightly earlier, but they're just, I think that's because they're just not getting enough of that cue, and the plain spadefoots are getting most of it. That's an interesting thought, and so there's a new postdoc in our lab who's looking at that kind of thing, and I just don't know. I mean, it's an interesting thought that there may be he's interested in personality things, and so that's kind of one of the things he wants to look at. Are they more exploratory, also as adults? I mean, this is something you're interested in, is how larval phenotypes translate over in adult phenotypes, and so it's very it's fun to speculate about that, and that is something that we're going to be looking at. Yeah.
Wanted to sort of play devil's advocate for a second. So with your plasticity first hypothesis, shouldn't there also be a null hypothesis that mutation first could also just as well give rise to the adaptation? And I'm just wondering, is there a way in your system by using like comparative genetics or quantitative genetics that you could test that the mutation initially gave rise to?
Yeah, so the question was, from the devil's advocate here, is that could you argue that a mutation first scenario, and are there ways of testing that? So, what West Eberhard was doing in that statement about, and I think it's really unfortunate that she did that, right? So it's kind of like Richard Dawkins with the selfish gene. We all know what he was doing, but it caused a lot of problems here, right? Clearly, you can't, it can't be plasticity first, because you have to have genes there that encode for the plasticity, right? And so there's, you could say you had to have that original cryptic genetic variation in the first place. So I totally agree with you. There had to be a gene first, if you want to put it that very, very simplistically. I think the point, though, that she's trying to make, and that I was trying to make today, is that there's no evidence that we've found so far that there is a genetic mutation that's occurring that's causing this, right? So it's consistent with this idea that a change in the environment is what is uncovering cryptic genetic variation that selection can then act on and start to refine. And obviously there has to be a lot of mutations then that, so you start producing a slightly shorter gut, right, when you eat shrimp. Now you wait around for those other mutations to start to occur, to make you a little bit better at getting the shrimp. So mutations to be able to see the shrimp better, to be able to digest that protein better, to be a faster swimmer, so tail muscles and so forth. So it's not an either-or situation, but the question is, the idea here is that the change in the environment and the plasticity might have played a more critical role in starting this process off. And unlike with a genetic mutation, this could potentially be happening again, impacting many individuals simultaneously, right? Whereas a mutation is going to affect one individual, and maybe it's direct descendants, right? And so you've got a population where suddenly it's like, wow, look at all this shrimp, and then they all start doing it, right? And then they're all exposed to that, that new cue, and you start evolution down that pathway, right? And this is where, so I don't think this is radically different from the way, that people think about how evolution and selection works anyway. And this is why I'm not going to be in the camp that says, we need an evolutionary synthesis, right? So that's not what I'm saying here. But I do think that it's a little bit different. Maybe it's just a little bit, but I do think it's at least a little bit different than sort of our traditional view of how adaptive evolution worked. I mean, I could be wrong, but that's kind of my thinking here. Yeah. Right. Right. Right.
And some of the things you said later in the talk made me think that there might be density dependence here. I wonder if you have any mechanism for thinking about how the initial slightly carnivorous spadefoot crosses that. And
yeah.
Possibility, I wonder if you've ever if you ever see pools of entirely omnivorous tadpoles and then introduce slightly carnivorous.
So that's a good question. So the question is, the disruptive selection data suggests there's a fitness valley for those intermediates, and that's a good point. And so that obviously, what I'm arguing here is they somehow had to get through that valley, and I don't know how they did that. And that's a very good point, because these I don't know what you call them, these sort of precursor carnivores we're getting are definitely intermediate. I mean, they're nowhere near being they're sort of more omnivore, honestly, than intermediate. But I don't know how it crossed that fitness valley. I mean, so answering the second part of your question, and so that's a really great question. I don't have an answer to that. But answering the other question you asked, so what we think is it's probably, you get a where is it? It's here somewhere. You do get